friends, welcome to Arnav's World. This is the 102nd episode in the biology series. Today's video is about a digestive juice known as bile or also known as gall. Yes, so it's a digestive juice created in the liver, secreted by the liver. So in the liver it is produced, 10% of it is stored in the gallbladder, which is normally depicted as green. So I'll use green. After which it is injected into the small intestine. First part known as the duodenum. So let's begin. In this video we're going to discuss about what this bile juice does and practically what it's made out of. So bile juice is made out of three main things. First, just mostly water. Then a few different acids known as bile acids. There are many different acids in this category and all of them help in digestion. And finally, bilirubin. It's a byproduct of cleaning blood in the liver. Yes, it's a byproduct which is created, which of course needs to be disposed of. So when the bile juice is being disposed of, like it goes to the large intestine and just goes out, the bilirubin is also taken out along with it. So now let's go into the main function of this bile juice. So let's take a small cross section of the small intestine. There are villi, thousands of villi, and on both sides, every side. Yes. So inside this villi there are many different veins, capillaries, lymph nodes, lymph veins. Yeah, think the blue is the blood vessels here. Okay, so normally nutrients which have been digested will come and be absorbed into the villi. Yes, and then be absorbed into the bloodstream where it be given to every cell which needs those nutrients. Yes. So yes, this is what happens after all of digestion occurs. But there are a few complications. There are a few exceptions. The main one being fats and lipids. Yes. So fats and lipids are particles which are too big to enter into the villi. Yes, and along with that they need to go through the lymphatic system, which we already talked about before, but we'll just going to continue talk about it later. So through the lymphatic system, because the fat molecules, even after they are broken down, are still too big to go through the tiny capillaries, because they might just block the capillaries. So blood will be stopped and you might get something bad. Yeah. So now let's just talk about the function of this gall or this bile. Yes, so let's begin. So fat goes into the, wait, I'll make an even enlarged model. So we'll use black for the small intestine and the villi. So these villi are very small. This is not for scale. Villi are not so close together in the small intestine compared to the size, which are just a few micrometers big. Yes. So these are the villi which we are talking about. So within the villi there will be fat molecules and many other nutrients going in. So this is a fat molecule and this is another fat molecule or just lipids. Fats all are counted as a type of molecules known as lipids. Yes. So other molecules such as waxes, cholesterol and many other things are counted in as lipids. Yes. So fat is one of the lipids. There are also other many lipids, but let's just continue. So these lipids will of course go through the digestive system. The bile juice will also be mixed in the fluid which is in the tract in the small intestine. So here there will be things called micelles. So these are automatically made. Let's just zoom in. So this is the zoom in. So this is the fat, the lipids. There will be an outer coating around these known as the micelle. It's like a container. You know, lysosomes or food vacuoles in cells. It's practically like that, but at a smaller scale and to contain lipids outside of the cell. Yes, in digestion. So, but how are these formed? They are formed automatically, completely automatically, because lipids and water don't like to be near each other, right? They will try to be as far as possible. So that's why they will create a boundary wall made out of a few lipids known as a few phospholipids per se. So these phospholipids are, okay, let's take this phospholipid. One side is waterphobic. That means it doesn't want to, hydrophobic. That means it doesn't want to touch water. It wants to be as far away it wants to be from water. Meanwhile, the other one is hydrophilic, which means it wants to be with water. If there's water, come, I want to hug you. Yeah. Meanwhile, the hydrophobics, the water, no, water, doesn't want it. So, both of these are of the same particle, a phospholipid. So one side doesn't like water and this one other side likes to be in water. So in here, this side likes water. It's hydrophobic, philic. While this inner side is hydrophobic. Yeah. So in this hydrophobic part also likes to be with fats or lipids. So the lipids would be stored inside these micelles. Yes. So then these micelles, as they are not like cells, they won't automatically go through there. But eventually these micelles would go to the villi. Yes. Now let's take a zoom in again. Okay. We zoom into this. Wall of the villi. Now we'll draw some stuff which is inside. There'll be many capillaries. Veins. These capillaries, these arteries will turn into veins here because they'll be carried in. Yeah. So and then also along with this, many people don't know this, but there's also a lymphatic system thing. Yeah. So like a vein of the lymphatic system, which is also sent in here to absorb the fats. So there are two main types of lipids in here. There are of course many other types, but there are two main types: triglycerine and these phospholipids again. Yes. So triglycerine cannot fit through this. It has a very big chemical formula. It literally has 98 hydrogen atoms and even 55 carbon atoms and many more atoms. So it can't fit through these tiny gaps which let these nutrients be absorbed into the villi. So they need to be broken down. So there is another enzyme or digestive juice known as, guess it, pancreatic lipase. So this pancreatic lipase, wait, I'll use blue. This pancreatic lipase comes and absolutely turns this triglyceride into monoglyceride, dividing it by 3. Yeah, so it converts into the triglyceride into monoglyceride and a few other free fats. Yes, so I'll just basically show how triglyceride is. So there's a main structure here and 3 main long columns. Yes, so in monoglycerin, these 2 are removed and they become free fats. Yes, so this is basically what happens. And monoglycerin, it can be absorbed, and these fats as well can be absorbed too. So the phospholipids, they can be absorbed into the villi by their own. They don't need to be broken down into smaller and smaller pieces. So as they go in, instead of the normal veins and arteries, they'll go to the lymphatic system, where these monolipids and free fats would again fuse into monoglyceride, when these other free fats would fuse together to reform the triglyceride. Yes, so they will just reform again because monoglyceride does not exactly have a use in the body, while triglyceride has many different uses, mainly in making energy. Yes, so yeah, triglyceride is nominated. So the lymphatic system is a bit more wider than the circulatory system veins. So that's why fat and this stuff could be carried through the lymphatic system because these particles would be too big for the veins and capillaries. This is just the main use of the bile juice. Yeah. How does this rhyme? The main use of the bile juice. Yeah. Okay. So, but there are also a few other uses. We need to rub all of this. Yeah. So, as I told before, another use of this is by acting as a medium for excreting the bilirubin, a useless byproduct which is secreted by the liver when just screening the blood. Yes, but this is one of the uses, one of the functions. But there are two more functions. No, sorry, one more function. Yeah. So, like the stomach, it will go here to the duodenum. So stomach acid, HCl and many other acids are acids. They have a lower pH level. Okay, so these have a lower pH level. That means they are more acidic. Meanwhile, this bile has a higher pH. pH is higher. We already talked about this pH scale in a previous videos in like physics and chemistry, but just watch those videos if you want to understand this acids and base stuff. But yeah, let's just continue this video. So while bile has a higher level, a bit like 8.5 or so. Yes. Meanwhile, these gastric acids have a very low amount, like 1.5 to 3.5, which is acidic. Yeah. So sometimes these acids can also enter the small intestine. So this bile will help to neutralize the pH level, neutralize the acidic medium, practically making it zero, which is pretty helpful. So yeah, this for today about bile juice. So bye.